All right, all set. It's nice to see you all again. I find it strange to start at 9:25. It seems like such a weird time to be here, but I'm sure it'll sink in just in time for the end of the semester. So in our last lecture, we did the sort of general first day introduction stuff. We went over the syllabus and showed you the Moodle site, everything like that. And then we started talking a little bit about some of the history that's going to be important as we start studying how behavior and the brain work together. And so we're going to continue on with that little bit of a history lesson. And you'll find that a lot of these names start coming up sort of multiple times. So we introduce them in the first chapter, and then when we get to the full chapter that discusses a relevant topic, they'll come up again, and we'll sort of remind you of what they did. And so one of these are going to be some of the individuals who started looking at electricity in the brain, and how electrical signals, rather than animal spirits, might be what's involved in getting us to move and think and do things. So we're going to start back in the late 1700s, and so you might have heard of Galvani, and he was famous for having used electricity to stimulate nerves. So he's the guy who hooked up frog legs to electrodes and got the muscles in the legs to contract. And he even found that if he moved those muscles outside of the leg, if he disconnected it from the leg itself, he could still make the muscles contract. So that's the first inkling that, well, okay, so it's not animal spirits or something akin to hydraulic pressure. It's actually going to be something to do with electricity. So maybe there's something in our cells that acts like a conductive wire. And so a little bit later on, in the 1870s, Fritz and Hitzig, there we go, I'm so bad with names, they produced movement by electrically stimulating the brain. So they had a dog, and by poking at different parts of their brain with an electrical stimulus, they could cause muscle contractions elsewhere in the body. And this is going to be really important when we start talking about sort of actions and parts of our brain being sort of localized. So different parts of the brain being responsible or controlling certain parts of the body. So we'll come into that in just a couple of slides. And then our last individual is Hermann von Helmholtz. And he demonstrated that nerves, though they on the surface look like they might be behaving like wires, having the electrical signal leading to contractions or whatever they're going to lead to, he found that the speed that action was occurring was much slower than what would be anticipated if they were just using a conductive wire. So on its own, electricity will travel fairly quickly in the range of the speed of light. But with these conductive nerves, the speed was much, much slower. And so that tells them that, okay, it's not just conductivity that's going on here. There's a biological process involved. And that started to give them hope that if it's a biological process, then we, by studying the body, can actually study that process and figure out what's going on. It's not going to be just electrons moving across a wire. It's actually something that we can learn and study and get more out of. And that brings us right into localization. And so localization is the idea that specific areas of your brain will carry out specific functions. And so there was a bunch of bits of pieces of evidence that led to this being their theory. So by electrically stimulating a dog's brain, they could cause movement in predictable places. They also had the famous case of Phineas Gage. As a reminder, he was the railroad worker who had a spike exploded through his frontal lobe, and it changed his personality dramatically. So he became volatile and stopped following societal norms. and they assumed that the damage to that specific part of the brain led to him having problems with those specific functions. And even Broca, the one who discovered Broca's area, had found that a man who had suffered a stroke and lost his ability to produce speech had damage to the specific part of the brain that we now call Broca's area. So there's a bunch of evidence that says, yes, there are parts of the brain that have specific functions. The problem here is that people started taking this sort of to an extreme. And the textbook does an interesting thing where they comment on it having been taken to the extreme, as a lot of things throughout science have been, and they caution us against doing similar in the future, which is a little interesting philosophy point there. But they talk about phrenology, which is basically a bunch of researchers who would document the shape and the size of your skull and assume, based on where the bumps and lumps and points on your skull were, what the shape of your brain underneath was. And so they did this fairly scientifically. They measured thousands of people's heads and then related the weird shapes they found to different personality traits or characteristics. So they said that if you have, a large forehead or something, it meant this. Or if you had very strong protuberances at the back, it meant that. But they started taking that, as we said, a little too far, and it starts to get to the point of there are some desirable traits and undesirable traits. And you could tell all of that from the skull, which is a little bit problematic, to say the least. And of course, the shape of the skull has almost nothing to do with the shape and the layout of your brain underneath. The bone itself doesn't hug the brain perfectly, so you can't tell what the brain's doing underneath. So it was more of a pseudoscience than an actual science. And then that led to a swing in the other direction, where people said, no, it's not that each and every point in the brain is responsible for a single thing. It's the other way. We have this equipotentiality, where the brain can function as a completely undifferentiated whole. And they argue that damage to the brain led to differing problems based not on the location, but on the severity of the issue. So Phineas Gage would have had massive problems because he had massive damage with a railroad spike. And so we can look at that now and kind of say that we're somewhere in between. It's not specifically localization, and it's not completely undifferentiated function. We're somewhere in the middle, where different parts of the brain will work together to produce a function. So we have a little bit of differentiality, but our functions are more diffused across the brain. And as we get into more topics in this course, you start to realize how complex it is to do simple actions. So even standing upright, that's going to involve the cerebellum for balance, and that's going to involve the motor cortex to keep me balanced. But it also involves perception of my balance and where I am to make those small adjustments to keep me upright. So what on the surface looks like a fairly simple act is using large strips of my brain and the back and everything in between. So we're going to be using something kind of in the middle of localized and non-localized, and we'll say that it's diffused but connected. And so the next couple of slides that we're going to go over should theoretically be a review of some of the biology and specifically sort of genetics biology stuff that you may have seen in the past. So we're going to touch on it fairly briefly here. If any of it's going over your head, I can always slow down and go into more detail, but again, first chapter is meant to give us a, hey, remember this exists? In later chapters, we're going to go more in depth. So if you need me to pause or expand, let me know. Move that so I don't trip. All right, so we're going to talk about the nature versus nurture debate. And this is one of the biggest debates that you'll ever hear people talk about in terms of biology or psychology, especially as it relates to people, but you can look at this for any species. And this is the ongoing debate about whether heredity, our genetics, is the most important thing, or whether our environment and our experience is the most important factor in determining who we are and how we behave and what we do. So nature being our genetics, our genes, what we are from what we've inherited from our parents. Is everything controlled internally? Is it rigid and predetermined? Or is it nurture? Is it all about how we're raised, the experiences that we have, and the environment that we're in? And so this is a fairly controversial topic. And I'm finding as time goes on, it's seen almost more controversial outside of the sciences, where people don't like the idea of genetics because it's too determinism-ish, and people don't necessarily love just the environment being responsible because you can see that you look like your parents, so there must be something genetic maybe. And in fact, within most sciences and most classes, there's a pretty healthy middle ground that most people can agree on. Most being the key word here. You will still get some debates going. But we tend to operate somewhere in the middle, saying that our genes, our nature, are going to give us sort of a span in which to operate. So they'll give us a reaction range where we can exist. So I always use the example of height because it's nice and easy to visualize. So my father is over 6 feet and my mom is just over 5 feet. So I was going to be somewhere in between the two. And then my environment, the nurture part of it, sort of what I eat and all the nutrients I get and things around me determine where in that range I fall. So I'm about halfway between both of my parents. I fell right in the middle of my reaction range. And we can think of these ranges for anything, whether it's a physical trait or even a behavioral trait. So maybe you could look at it as activity levels. How much energy do you have? Maybe there's a range from your genes as to how much energy you might have. And then your environment is going to influence the genes that you have to determine where you fall in between. So our take home here is that while we talk about it as nature versus nurture, and it's a debate of one side versus the other, the real answer seems to fall somewhere in between. And we'll start talking about different traits have sort of a lean one way or the other. So things like height might be a little bit more genetically influenced. If you have the genes to be tall, you're a lot more likely to be tall. But there are other things that are a lot more environmentally influenced. So some of them are, even if you have good genes, maybe your environment is a stronger factor. And we can actually quantify that and talk about how much of a particular trait is influenced by either genes or environment, kind of as a ratio. And so as that becomes relevant, we can talk more about it. And so we've talked about genes, but we haven't really defined them or talked about how they exist or what they are. So genes are our biological units, and they're going to direct all of the cellular processes, and that's how we transmit or pass along traits between generations. So genes are segments of our DNA, and they are basically sets of amino acids, pairs that are going to tell our bodies what to do with information that they encode. So most of the genes that we think of, most of the functional genes that we care about, are what we would call protein encoding genes. And that just means that these genes, when our body translates them, when it makes use of them, it'll turn them into a protein through a lengthy process that we don't care too much about yet. But it'll turn it into a protein, and those proteins can go off and do things. So you might have a gene that's responsible for pigment color in your hair. And so whenever your body is growing hair, it might say, oh, it's time, we need pigments to mix in with this. And then those genes will activate and produce proteins that are that pigment in your hair. And it's not always that direct. Sometimes it'll produce proteins that turn on other genes, and those genes turn on other genes. And sometimes they make proteins that then work together to do something. But for the most basic point, we keep it to the genes eventually get turned into information for our proteins, and then the proteins do stuff. So everything in our body is regulated, at least in a large part, by our genes. And for DNA, that's deoxyribonucleic acid. Hopefully you guys know that already. But it's just double-stranded, double helix chain of chemical molecules. That's when you look at the strings of A's, C's, T's, and G's, and they pair up accordingly. And the order of those amino acids the order of those bases will tell us how to read them, turn them into amino acids, and turn them into proteins. All right. So continuing on with stuff like this, the textbook does a really good job of giving extra details if you want more detail, but they also throw a lot of topics at us this chapter, which is why this seems so scattered. But it does give us a good background for most of the things we're going to need here. So with this whole genetic code thing and the idea of passing on or inheriting genes between generations, we also probably want to talk about the process where you pass along genes. So when we have DNA coming from a mother and a father and they fuse together, they form a zygote, which is a fertilized egg. So egg plus sperm gives us a zygote. And what this works is each parent contributes 23 chromosomes in humans. Different species have different numbers of chromosomes, but we care mainly about humans here. So you get 23 from one parent, 23 from the other, so that when they fuse, a full organism will have 46 chromosomes. And so they're paired off. You get 23 pairs to get that total of 46. And the idea is that one in each pair comes from each parent. And so that will eventually sort of grow and develop and become a full organism that has the genetic material that they received from their parents. And they divide up what we call this organism as it goes from a fertilized egg off to an infant when it's born. So starting as a zygote, as it starts developing, as it grows, it turns into what we call an embryo. So that's over the first 8 weeks after fertilization is when it's in the embryo stage. After 8 weeks, we call it a fetus. And this is just sort of developmental terminology. They chop it up that way. All right, and back to genes, now that we're talking about sort of putting genes together, having them come in from our parents, we can also say, because we have these different pairs of chromosomes, assumedly, if each chromosome has a bunch of genes on it, because our genes are all on our DNA, or genes are DNA, then we will have different versions, potentially, of those genes that we get from each parent. So we can call versions of a gene alleles. And this is the genetic terminology here. But if we go back to that example of hair pigment, maybe your father is blonde and your mother has brown hair. So they would have different alleles or different versions of the gene that encodes hair pigment. And so what we might have is one of those alleles would be dominant, meaning that it's going to be producing an effect regardless of which other allele it's paired with. So brown hair is usually on the dominant side of things because it's going to be producing lots of dark pigment. Whereas blonde would probably be a recessive allele. And recessive alleles are only going to produce influence if they're paired with other recessive alleles. And the best way to think of this is if you have that brown allele, that's the dominant one that's going to tell the cell to produce lots of brown pigment. The blonde allele would actually probably just be a broken version of the brown allele. So instead of producing a different kind of pigment, what it probably does is produce no pigment or very little pigment. And so we have a dominant allele that's saying brown, 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 and then we have the blonde allele that says nothing. And so we end up with brown hair. If we had two recessive alleles, both parents are blonde, they both pass on that blonde gene, Neither of them tells the cell to produce pigment, so you end up seeing that blonde trait, but only because there isn't a dominant allele present. If we have dominant alleles, even one copy of a dominant allele is going to usually be enough to see that particular trait. But the recessive alleles, you need two versions to see it because they get washed out by the dominant alleles. And this is a very simplified version of this. If you go into genetics, you talk about weird, either there's a halfway in between or they share dominance. Things get way more complicated. But for this class, we stick to very simple, one or two alleles at a time kind of thing. But if you have taken those classes, you know that this gets a lot more advanced. And so if we wanted to talk about that arrangement of alleles, we could say that our first example, where we had a brown and a blonde allele, we would say that you are heterozygous, so you have one of each version. If we use the other examples, where I said you had two of the recessive blonde alleles, we would say that you're homozygous recessive, meaning that you have two identical alleles, and we're specifying that both of them are recessive. When we were talking about heterozygous, each allele is different, we don't have to specify in this environment because we only have two that we're looking at, a dominant and a recessive, and if we're saying you have one of each, then you know you have one of each. If we start getting into those more complex systems, you would see heterozygous, and they might use nomenclature to specify what that arrangement is, what the two different versions are. And you can also be homozygous dominant. So instead of having two of the recessive blonde alleles, maybe you have two of the dominant brown alleles. So you could have that, and that would work just as well. That could be called homozygous dominant. So that's some terminology that might pop up as we talk about these different alleles, which versions of a gene you have. And a great example of this is blood types. This is an interesting choice for the textbook to go with because instead of my example of blonde and brown, where I give you sort of one dominant and one recessive, blood types are one of those complex situations where we have multiple dominant alleles. So with blood types, it's not just one dominant and one recessive. We actually have two dominants. So A and B on this chart show up as capital letters, and that's the standard notation to indicate a dominant allele. And so what that means is if you have an A allele, it's telling your cells to produce those A proteins. And with the B allele, that's telling your cells to produce B proteins. And our recessive, lowercase o, is saying don't produce proteins. So it's a non-functional version. So if we look at our parents, so our type A parent genes at the top there of the chart, we're saying that our parent has two alleles. They got those from their parents, and this chart is showing what they pass on to their parents. So let's just start at the top. And so we have a capital A and a little o. And what that tells us is that they have one copy of the gene that says make A proteins, and one copy of the gene that says don't make proteins. So what we're going to see, the phenotype, the trait that we observe, is going to be blood cells with A proteins. And then if we look at the side, we have our type B parent genes. And they also have two copies, two versions of their gene. They have a capital B and they have an O. So for them, they have that one dominant capital B that says make B proteins and the little o that says don't make proteins. So it's going to overall look like B proteins because they have at least one dominant gene that's saying to do that. So those are our parents. And then what this chart is showing us is what they might pass along to their theoretical children. So these are Punnett squares, and if you've done them, you know them very well. If you haven't done them, they're just a simple representation of which versions of a gene parents pass down. So in our first column, we have our capital A at the top. So we're saying that parent number 1 is going to pass on a capital A allele, and across the top that way, the same cell in the middle there, our second parent is going to pass along a capital B. That's what our probability is going to give us for that one potential offspring. So if parent 1 gives an A and parent 2 gives a B, then we have A and B alleles. So from what we understand here, where both of those are dominant, they're both going to tell the cell to produce their respective proteins. So somebody who has an A and a B allele will have cells that produce both A and B proteins. So they are type AB blood. If we go across that top row, we're going to say that our first parent, instead of giving a capital A, this time, by chance, they're going to give a little o. So they're going to pass on an O allele. But the second parent is still passing along a B allele. So now our offspring has the genotype, has the genes, the alleles of capital B and little o. So they have produce B proteins and they have no proteins. So what we see, our phenotype, is that they have type B proteins. And then if we go down the outside, where our first parent gives a capital A, our second parent gives an O, we have type A, where they produce A proteins and then produce no proteins. So what we observe is type A blood. And then the cool one at the very bottom, the last one, is that each parent gives that recessive O, and the type that we see is type O. There are no proteins produced, so they are the recessive. And so here we're seeing two dominant alleles that can work together, and then the one recessive allele that is only ever seen if it's paired with another recessive allele. So that's a pretty good example of that. And it might be a good idea to understand how these kinds of charts might work. Maybe to practice, think about what would happen if one of the parents was type AB instead of AO, or something like that, and how does that change the potential offspring that they might have. So this is kind of an interesting exercise to practice if you haven't spent a lot of time with Punnett squares. If you have, then you are probably well versed in them and sick of them. But I've been sneaking in a little bit of terminology as we've been talking about these things. So this is where the actual definitions come in. So I mentioned that if we have a parent with a we have an offspring that has a allele and an O allele, the observed trait, the phenotype, is type A blood. So our phenotype is what the genotype means in reality. It's what we see when everything is put together and everything is acted. So the phenotype is that observable characteristic. It's what we can look at. So with my example of brown versus blonde hair, the hair color that we see at the end would be their phenotype. Is the child brown-haired or blonde-haired? The genotype is looking at the specific combination of alleles. So what versions of each gene do they have? So if I specify that somebody has one of the A alleles and an O allele, their phenotype is still going to be type A blood, but their genotype is A and O. We can have another individual that has two copies of A. So they have A and A. Their genotype is therefore different because they have two A's now. But the phenotype, the observed blood type, is still just type A because that's all the proteins they're producing. So your genotype can be different or give us more information than the phenotype does. And our phenotype doesn't necessarily tell us exactly what our genotype is. So there are multiple ways that we can have a particular phenotype. So the example with type A blood, we see the phenotype of type A, and you could have A and A, or you could have A and O. And that works with B's as well. So there are ways that different traits can be hidden. So just keep in mind that our phenotype isn't always going to tell us what our genotype is. And our genotype is basically what happens when the genes and the function of the proteins that those genes encode is translated and produced. So our genotype plus the environment of our body and what's around us is what ends up producing our phenotype. So genotype plus environment equals phenotype is a good thing to remember. And then we can talk about some of the more complicated cases that we might run into, because of course our examples have been fairly simple, but we can talk about things like X-linked traits. So most of what you'll talk about, if they don't specify that it is X-linked or sex-linked, then we're talking about our autosomes, which basically means everything that isn't an X or Y chromosome. And the autosomes work really nice and normally because they're the exact same whether they come from your father or your mother. But our X and Y chromosomes are a different case, which is why we're talking about them now. And so what happens here is that for our sex chromosomes, an X chromosome is kind of just like a normal chromosome. It's nice and big, it has lots of space, and therefore lots of genes. But the problem is that the corresponding other sex chromosome, the Y chromosome, is much smaller. And so the Y chromosome has much fewer genes on it, and therefore, if you have an X and a Y, you might not have two copies of all of the genes that are contained therein. So an X chromosome has a lot more genes that don't have a match with the Y. And so a really good example of this is something like color blindness. If you have ever looked into color blindness, you might have noticed that it is very common in men, but not in women. And that's because it's an X-linked trait. So if your father is colorblind, that means that his X chromosome, the chromosome that he got from his mother, has a gene that doesn't allow him to see colors normally. And the Y chromosome doesn't have a matching gene to balance that out. So color blindness is recessive, but because the Y chromosome doesn't have an equivalent gene, you see the recessive copy because there's nothing else to balance it out. Whereas if a female if they had color blindness, they would need to have two copies of that gene that produces color blindness. So both of their X chromosomes would have had to have the exact same non-functional recessive gene to produce the same thing. So it's a lot more likely for someone who has XY chromosomes to have that colorblind trait because there's no protection offered by the Y chromosome. Whereas for somebody who is XX, they need to have two copies of the same gene or the same version of the gene, the same allele, to produce that same problem. And so if you ever look at distributions in population, and there's a severe disparity between if XX individuals and XY individuals are affected, you can usually indicate that it's an X-linked trait. There are also some really cool cases of Y-linked traits, but we don't really talk too much about those because they're very, very rare. What is a lot more common is talking about things that are polygenic. So I've, again, simplified the topic, and we're talking about sort of a gene that determines the pigment of your hair, or a gene that influences whether you can see color or not. But most of our traits of interest, and especially as we get into talking about behaviors, those things are going to be polygenic, which just means that they don't use a single gene, they'll draw from multiple genes. So these characteristics, these behaviors, these traits are determined by several genes. So maybe we have to look at two different gene locations, and we have to know what alleles we have at each of those locations to determine how they interact. And a really good example of this one is actually skin pigmentation. The same way we talked about hair color, produce lots of pigment or produce a little bit of pigment. Skin pigmentation is controlled by 6 or more different genes that each will say produce more or less pigment. So if you are very pale, then at each of those 6 locations, you have alleles that say no pigment. Whereas if your skin is darker, at least some of those locations, you have alleles that say produce more pigment. And so the combination across all 6 of those locations determines how much pigment overall is produced, and that affects how dark your skin is in the end. And so we can break that down across multiple different kinds of genes, and especially with behaviors, we find that it is very difficult to find a single gene that is a determining factor for most behaviors. And in fact, it can get complicated enough that we'll say that, there's 20 possible locations that may or may not be involved with this particular behavior, and it might differ between families, because that's how looking at behaviors in a genetic sense ends up going. But to get started on trying to figure out which genes might actually be related to different disorders or behaviors or traits, you kind of have to know what genes there are. If we wanted to run a study and look at individuals that have a particular disorder, or maybe we want to figure out where colorblindness is on the chromosomes, we could get a bunch of people who are colorblind and a bunch of people who are not colorblind, and we could compare their DNA. We can see if there's a particular mutation that those who are colorblind have that those who aren't colorblind don't have, or vice versa. And if we can find things that are different between people that have a particular trait and don't have a particular trait, that gives us a pretty good indication of where to look, if we have a map. And that's where the Human Genome Project came in. A genome is all of the genes on all of our chromosomes. So it is a map of all of the human genes. And there are genome projects for lots of different species. Mostly people started with common lab species. So lab rats, mice, Drosophila, nematodes, all the really common model organisms, those would have been sequenced first because most of their genomes are much smaller than ours. The human genome is a massive project. And their intent with this project was to try and map the locations of all of the different genes on the human chromosomes. So they wanted a map of all 23 types of chromosomes with our pairs and know what kinds of genes might be found on each of those chromosomes. And they were doing this to try and then make connections. When we were talking about turning our genotype through all those processes, through the environmental interaction into our phenotype, that's gene expression. That's the process of turning what we have on our genes into what we can then observe later on. It's going to be expressing what's in those genes. And this project took a very, very long time. They reported their first completed, in quotation marks, stage in the year 2000, because that was supposed to be a big revolutionary thing. It was the 10-year anniversary of starting the project, and it was a very convenient year. And then 5 years or so later, they said, okay, we've actually finished. So they announced that they were done with some blanks. They're like, it's mostly done. And then they still took 5 years to get more copies, because you can't just sequence a single individual's genome. That doesn't give you a good picture of humankind as a whole. So they'd have to sequence multiple people. And I believe when they started, they had at least 20, and over time, that gets bigger and bigger and bigger. And the really cool part here, this is just a little bit of an aside, is that technology at the beginning was really, really slow. So DNA sequencing at the time was a very manual process. If you've ever done a bio lab where they have you mix in different amino acids, different bases, and slowly build up different copies using PCR, you can actually, one by one, identify each base in order, and it is an exhausting process. And so it used to be that in the 90s, you could get an entire PhD from sequencing a single gene. Nowadays, you can sequence a single gene in an afternoon. And most universities actually have a machine that will read out the order of DNA. So the technology has moved forward in leaps and bounds. And nowadays, it's a lot faster, a lot easier to do that. In my own undergraduate research, when we were looking at the genes of single-celled organisms, we had to send out our DNA samples because there were labs that had that technology. Ours was not one of them, so we just sent ours to somebody who had it and we would pay them for their services. But it's gone quite a bit further since when I was doing this, partially manually and partially sending it out. And as stuff gets done faster, we're seeing a lot more progress in what we're finding and how detailed things can be. And because now it's so much easier to sequence genes, instead of having to wait years to identify where a particular gene might be located, we're starting to do it a lot faster. Which, again, could be taken to an extreme if people use a more deterministic perspective on genetics. If they perceive genes as determining exactly how you're going to behave or what you're going to do, that can be problematic. But we're going to use more of the idea of, instead of a deterministic perspective, more of what I've already explained as your genes setting a range in which you can fall. So we can talk about genes conferring vulnerability, basically where they give you a predisposition for a particular thing. And so if we're looking at behaviors, usually we're looking at psychological disorders. So we talk about a vulnerability as a predisposition for a disorder. But, kind of like my earlier example, even though your genes say that you have an increased risk of developing a disorder, it isn't a deterministic factor. What usually happens is there also needs to be particular stressors or particular stimuli from your environment that interact with those genes to produce the phenotype of having that disorder. So we're not on the fence of deterministic genes. There are very, very, very few disorders that are sort of hard linked to genetics. Off the top of my head, I think the only one I know of is Huntington's disease. And that's not so much a psychological disorder as a disease, but it is one of those where if you have a particular allele, you have the disorder. Yep. Yeah, and it's actually, this is a little bit more than unique, because I teach a class on genes and behavior, and half the class is talking about schizophrenia, because it was one of the first things that they were able to link genetically because it's common in families, but it also has like an 80% heritability, which is remarkable. So if you look at individuals who have family members with schizophrenia, their likelihood of having it themselves is like 10 times higher than someone without a family history. So it's a massive one, but it's not 100%. It's not completely deterministic. And as we get away from, or as we move towards sort of more abstract things, so things that are a little bit more general, like say anxiety, it's really hard to find a particular gene or even set of genes that everyone with anxiety has, that no one without anxiety has. So we get much smaller percentages in terms of heritability with other disorders. And it kind of makes sense because a lot of these come from different sources. Even that not quite 100% with schizophrenia comes from the fact that there are multiple families that have completely different mutations that have led to schizophrenia. So you might have one family group that all have schizophrenia from one particular gene, and another group that have schizophrenia from a completely different gene. So it's a lot of complex interactions, and I spend an entire semester talking about how inaccurate of a science it can be. Yep. Oh, that's an interesting one. This is just sort of general discussion because it's cool. But so sometimes there are like predispositions to categories of mental disorders. So the more you get into it, the more you realize that things like anxiety and depression and all the subcategories under there, they tend to be comorbid, which means that they occur together a lot more often than they occur separately. And so sometimes being predisposed to one kind of gets the other one tagged along. So you can end up with maybe because of your environment, maybe you were protected because you have good coping skills or a good environment. Maybe you were protected from the anxiety side of things, but the depression side of things wasn't as protected, so you developed that, or vice versa. So it's a very complicated moving system, and quite a few psychological disorders, as we learn more about them, we realize that they are very comorbid. There's a lot of sort of carry-on and overlap, and it's getting harder and harder to draw a harsh line between different categories, which is why a lot of people don't love the DSM-5 and the idea of hard categorizing disorders. Sorry, can you say the first part again? Yeah, and that's why, so when we talk about, say, like an 80% heritability in schizophrenia, we're not necessarily saying like 80% of it is coming from genetics and 20% from the environment, but we are saying that what we observe from the traits is strongly driven by the genes, and there is a smaller portion that's driven by the environment. But as we have lower heritabilities, we're saying that there's sort of less of a genetic tie and more of an environmental influence. Cool. Yeah?
This is kind of a more of a question, but I know Gordon's a popular article believes that there's no such thing as an irritated personality trait. And I'm wondering what.
Ooh, interesting. I don't know, because it's like, even though I have taught a class on genes and behavior, my expertise lies in non-human animals. So I don't really deal with easily classifiable things. So for me, if I'm looking at, say, anxiety traits in an animal, we're looking at things like decreased activity or hiding behavior or sticking to the walls. So it's a much more simplified model. And what I've found from dipping my toe into the topic is that I think it's a lot more complicated than we could ever adequately describe. And the categories are often very misleading. Yeah, so kind of a non-answer answer. But yeah, needless to say, I really enjoy these sort of topics. I find it very interesting, and that's where my background in genetics kind of carries forward into being useful talking about behavior. But this sort of stuff is really interesting, and it can get far more complicated than we need it to if it happens. Okay, so I even have heritability here. And they went with the simplified definition, which is the percentage of the variation in a characteristic attributed to genetic factors. So it's a little bit more complicated to that, which you might have surmised from my explanation. They don't love to say that it's a hard, if we say 80% heritability, it's not 80% genes and 20% environment, but that's the simplified, that's the level we want for this textbook, so I guess we're going to go with that. I basically like to look at it as if you have a higher heritability, more of that particular trait is being driven by the genes, and if you have a lower heritability trait, then it's mostly driven by the environment. And most things, especially when we're looking at behaviors or even psychological disorders, we're going to stick in the range of like 30 to 40%, which is why schizophrenia being as high as up to 80% in some estimates is something worth talking about. So there are special cases and individual disorders that have to be studied in a special way because they are so different. But for the most part, we deal with less defined terms. And so we've kind of already been talking about this idea of natural selection, but not without actually naming it. So we've been talking about passing on genes and having genes interacting with our environments to produce physical traits, things that we can observe and document. And this whole process is central to the idea of natural selection. And this is the idea that people or any organism can have genes that produce useful phenotypes. And having a useful phenotype means that they live longer and are more likely to reproduce. And because they live longer and reproduce more, those genes, those beneficial genes that led to the beneficial phenotype, get produced more in that population. So over time, natural selection leads to an increase in those useful genes, and it gets passed on more and more. And so we can say that the population is evolving because the proportion of different alleles in that population are changing. So as those useful phenotypes come into play, as we get more and more of the helpful alleles, the helpful versions of those genes, they'll become more prominent and the population changes overall. A really good thing to point out here is that natural selection can lead to something being more beneficial in a particular population at a particular time. This is again that idea of taking things too far, and some people will label certain alleles as good and certain alleles as bad, and especially with humans, we can go too far and start saying that things are desirable or undesirable, which again is problematic. So when we look at natural selection and evolution, we have to keep in mind that these traits are beneficial for this particular species at this particular time because of the exact environment that they're in. So if we use the example of plants, maybe you have one species of plant and they're spread out all across the city, but one part of the city gets hit with a supervillain's ice ray and it's really cold all the time. So now the plants that are really good at surviving in the cold are going to live and reproduce, and over time the cold part of the city will have plants that are really good in the cold. But if that ice ray goes away and it warms back up again, they're now adapted to living in the cold, and it isn't necessarily cold. So what was a useful or quote-unquote good adaptation is now not so helpful. So these things can shift based on the environment, and that's good to keep in mind. Oh, I forgot I had this entirely. So we were talking abstractly about heritability and seeing how related to genetics different traits are, and these are actually a bunch of really common behavioral disorders and medical disorders. And so this isn't exactly heritability on these axes. What we actually have is twin concordance. And so it's an approximation. So if you think of the fact that, how do we start this? Okay, step back and we'll talk about types of twins, and then we'll come back to the graph. So you might know that there are identical twins and fraternal twins, or homozygous twins and heterozygous twins. So you can have twins who came from a same fertilized egg that then split and became two people with identical genetic material. So those are our identical twins. And because they came from the same fertilized egg, they have 100% more or less of the same genetics. Whereas our heterozygous fraternal twins, fraternal meaning like brothers, they actually came from two separate eggs that were fertilized at the same time. So instead of being 100% identical, they only share as much of a genetic component as normal siblings would, which is only 50%. So we have 100% same genetics and 50% same genetics in these different kinds of twins. And that gives us a really cool situation where if we make some scientific assumptions that twins being raised in the same house by the same people should share quite a bit of their physical environment, we can then assume that any differences that we see between these identical twins and fraternal twins should be mostly driven by genetics and not from their environment. Yep. Actually, it's usually because of misidentification. So if you have children and they are two boys, or if you have twins and they're two boys, people assume that they're identical, but they don't have to be. So identical twins will always be the same. They'll either have XX or XY. Whereas our fraternal twins, we have kind of like that Punnett square, where if your mother is always passing down an X and your father has an X and a Y, half of the time children will be produced getting a Y, and half of the time they get an X from the father. So there's a 50-50 chance of boy or girl. But usually what happens is if the children are born and they are the same, there's an assumption that they're identical. And that isn't always the case. And that's very problematic in this kind of research. It causes a lot of problems. But, so if we have that assumption that, okay, so identical means all the genes are shared and fraternal means half the genes are shared, and we're assuming, it's a big assumption, equal environments, then any difference that we see between our blue and our green here, blue being the identical twins on the left, and green being the fraternal twins on the right, the bigger the difference there, the more of a genetic component to that particular trait. So if we look at sort of the biggest examples, we have, say, major affective disorder in the first graph here. There's a pretty big difference between our blue and our green bar. So we're saying that we have twin concordance. So the higher that is, if you have an identical twin with that disorder, you are that much more likely to have it. So a twin concordance of 100% says that if twin A has it, twin B always has it too. So these are looking at probabilities. Whereas if there's only a concordance of 0.2, that's only a 20% concordance. So if twin A has it, twin B only has a 20% chance of having it as well. So that's just what we're interpreting here. And what we get from it is that if we have a really high concordance for our identical twins and a fairly low concordance for our fraternal twins, we're saying that there's probably a pretty big genetic component there. If we see a really high concordance even in our fraternal twins, that's telling us that it's probably not the genetics. It's probably from being raised in the same environment. But you'll notice that there are quite a few that are fairly high, but only for identical twins. And that gives us some evidence for a genetic link for a lot of these things. Again, not a deterministic thing, because even the highest of these bars is only about a 60% concordance rate. So always keep in mind the scale of these when you see them, because it can be a little bit terrifying if you think about it too intensely. So hopefully that makes sense after we took a couple of steps back to talk about what it is we're looking at. And as we come across these things, I will do sort of little asides to make sure that we know what it is the graphs are telling us. All right, and that brings us perfectly to our last slide, which is sort of a big summary of what we've talked about so far. And one of the biggest take-homes that I'm hoping you're going to have gotten from this is that studying the brain is still, they call it a frontier, basically meaning that there's a lot of unknowns. We are just getting to the point where our technology allows us to study a lot of the things that people have been interested in for centuries. And as the technology allows it to happen, we're getting to learn more and more. And as I said, the more we learn, the more we realize that we don't know. And a lot of the answers that we get, as disappointing as it is, are we don't know or we're not sure. So even in our textbooks, they'll give us, oh, it might be this, or it might be this. And there's evidence for both, but also for neither. So you kind of get used to that almost disappointment of not knowing. And then hopefully, as more research comes out, we'll get closer and closer to understanding how it works. And so if we're talking about doing well in the field of sort of brain and behavior studies, then we can start looking at things like we're building on the discoveries and the thoughts and the ideas and the groundwork laid by the predecessors in the field. And because psychology is fairly new, we're not just building on the history of psychology, we're building on the history of other fields as well. So we brought in our philosophers, our physiology experts. All of that leads to what we know and work from today. As I said, the developments in technology are massive. And as these are moving, and they're moving fairly quickly because they're happening in my own lifetime. I went from having to ship out gene samples to you can do them in your own lab. And it's actually at the point, last time I checked, you can get a DNA sequencer for only like $50,000, which is a large sum of money, but it's not unheard of for a university to invest that, to not have to send all of your samples elsewhere and wait months to get results back. It used to be that you would spend in the ballpark of $1 or $2 million to get the same technology. And now they're much smaller and a lot easier to use, so massive advancements. A big portion of getting to where we are now evolved from that change from more of a subjective, sort of fluffy interpretation of things to a more empirical, scientific perspective, and having that approach of critically evaluating our environment, having a systemic approach to gathering evidence and evaluating it critically. That's all very important. And we also have that issue where we've kind of agreed to not know how we're going to handle the mind. We've said that a lot of people, for ease, will just assume materialistic monism, that the mind is our interpretation of the physical stuff happening within our brains. It's the weather of our thoughts, and we're going to try and see if that continues working throughout the course. But a lot of people will, if you go to a conference and they're in between sessions, they might debate the nature of the mind, but we don't expect to really solve that problem anytime soon. So we just accept and come to terms with the fact that we might not know what the mind is, and that's okay. But that gives us a lot of information, hopefully a lot of it being review, that'll set us up to do well in the coming chapters as we dig more into some of this info. So I will release you guys today, and I will see you next Tuesday.